Hello, this is a video on differential staining. This is under the topic of microscopy as a diagnostic tool. In your lab manual, it's not titled differential staining, it's titled gram staining. And so this is a bit of a tweak, so to speak, okay, because I wanted to cover both differential stainings, which is gram staining and Zn stain, acid fast staining. So just take note of that, please. You do not have the gram stain in the main lab menu, but I hope you have the leaflet with acid fast stain notes. Okay, so differential staining, whichever type it is, whether gram stain or the Zn stain, is put into steps. It can be summarized into 3 steps. Okay, and so you have your primary stain, and then you have your alcohol step as well, and then you have your counter stain. So you can see in my view here. Oh, please, by the way, bear with this amateur videography, but I do believe it to be effective, and I will get better. Yeah. So I was saying, this is as you can see in my view here, I just put a note that if it's gram stain, we have the use of a mordant in the gram stain, okay, which is iodine, but I'll just explain that later, a bit later just now. But other than that, these are the basic steps that we will see whether it's gram stain or Zn stain. So your primary stain, your alcohol, the decolorizer, and the counter stain. So what is gram stain? Gram stain is an initial step in bacterial investigation or identification, sorry, of a patient specimen. It's the most used method of trying microscopy method rather of trying to identify bacteria. Okay. There's a note there in my view that cultures are made alongside gram stain. So gram stain is not a standalone diagnostic tool. So do note that, please. So gram stain, as we said, is a microscopy tool, and it's the main purpose of gram staining is to differentiate the bacteria into gram positive bacteria and gram negative based on cell wall properties. Okay, so you've learned about this in your lecture notes. Recall those details of your gram negative and gram positive. Okay, the peptidoglycan. Hope I said that right. So we are using that principle to differentiate our bacterium into those 2 classes, or that distinct feature rather of bacterium. Bacterium. So you have in your lab menu, the main lab menu, so to speak, the steps of how a gram stain is done. Okay, so you can review those steps, but I will run through with a small illustration on those steps. Okay, but please read the steps alongside or after the video, whichever you prefer. Before my illustration, I just want to show you again those summarized steps of the differential stain gram stain. So I have my primary stain, which is my crystal violet, which is purple color. I have my alcohol, my decolorizer, and then I have my counter stain, okay, which is saffronin, which is pink. Sometimes you can take red as well. Saffronin. Okay, so those are my stains that you can see, and my alcohol there in the middle. There is a mordant in between 1 and 2, but let's just look at the illustration to just consolidate the steps together. Okay. Okay, so you can see in my view here, I have on my laptop screen the steps. Okay, so you have your bacteria smear, nice dried slide, okay, of whatever it is. Maybe it's a throat swab, maybe a CSF, the smear of it, whatever your specimen is. So you have made a nice dry impression, a smear, and then after that we apply. We do our we take we start our procedure, our gram stain procedure, right? So we have the first step application of the primary stain, the crystal violet. Okay, you can even see a nice purple in my screen there. So we have that applied, and then of course rinsed after a few minutes, or is it a minute? Your manual shows the exact time there. So you rinse it off after the specified time, and then we apply the mordant, like I said, in between the primary stain and the decolorizer. We have the mordant iodine, which makes a nice big complex of the crystal and iodine in the procedure of gram stain. So at this point, whether the bacterium is gram positive or gram negative, they are both taking in the crystal violet into their cell wall, into their cells. So the working definition of what is gram positive is if the bacterium retains the crystal violet, the primary stain. That is the working definition of what is gram positive. Okay, so as we move on to the decolorizing step, okay, where in this case we have ethyl alcohol. So if the bacterium in the sample is gram negative, this decolorizer will be able to penetrate that thin peptidoglycan and rinse out, so to speak, the crystal violet. Okay, so the gram negative at this point has not retained the crystal violet, has been rinsed out. Okay, it has been rinsed out. The gram positive, on the other hand, at this stage, when we apply the decolorizer, will retain the crystal violet because of that thick peptidoglycan. Okay, there is a nice structure there that kind of only dehydrates. It cannot really remove the crystal violet, only dehydrate the peptidoglycan. Okay, so I don't know, I hope that's clear there. The crystal violet is retained rather in the gram positive when we apply the decolorizer because of that thick cell wall. Okay, so anyway, at this point in the decolorizer, we would have, in the steps, sorry, at this point we would have gram positive bacteria if it was a gram positive specimen, and if it was gram negative, we would have colorless bacteria on this step. So that's why then we have the counter stain of safranin to apply in a gram negative slide, to apply to, regardless, anyway, you never know what it is. So regardless of what it is in the smear, I was just assuming, for example, that I had a gram negative bacteria in the slide where I only had colorless, and then I would apply my counter stain to give me color to my gram negative, right? But regardless of whether it is, like I've just said, whether it's gram positive, gram negative, we will apply the counter stain, okay, in the procedure. So that is the basic principle of gram stain. And the practice in the lab session is to appreciate these staining colors, and also the morphology of the bacterium themselves, the shapes. Okay, in our illustration here we have illustrations of cocci and rods. Okay, and then also arrangements are also part of the exercise in the lab session. Are they arranged in a particular way? Are they diplo? Are they in clusters and so on? So that is what our exercise is about, really. Okay, in the actual microscopy stations in the lab. So I have for you here, you can quickly appreciate how the microscopy view would actually look like and actually looks like. Okay, um, some previous sessions, we had some nice clusters like this, clusters and cocci. And here, of course, it's the purple. You can see the gram positive. You can see that identification with a nice purple. Then you have your pink coming up here with rods. I don't know if you can tell if there's a rods in the view, but you can appreciate something. But this is the exercise that you're expected to do, describing the microscopy view. Okay, the shape of the bacteria, the color, the arrangement. Okay, gram stain results here.